All right, guys, welcome back. So today's Monday, September 20th. I just posted kind of the schedule for the next 2 weeks in class, just so you guys know where we're going. You just had your second lecture exam, and the grade that you have entered, when I went through, there was a question about the rule of nines, which I don't think I know we didn't cover it in lecture, and I don't even know if your book covers. So if you got that question wrong, I gave you 2 extra points for it, because each question is worth 2 points. If you ever want to go through your exam, usually lab time is a good time to do that. Or if you don't have me for lab, if you can get to my office hours on Thursday, or even after class today, if you ever want to go over an exam, I'm happy to do that with you guys. So here's the schedule. Not for once, you guys don't have any tests coming up in this week or next week. You do have several lab activities though. So just make sure you're coming to lab so you don't miss out on any of those lab activity points. And then the last thing, I've heard from one of you who want your lab kit mailed. So in October and November, there are dates when you'll need to go to campus, pick up a lab kit, but we will mail them out to you guys who live a little bit farther away. And here are the lab kit pickup dates. You guys can go, or someone can go in your place. If you can't go, or you have an emergency situation, please email me your home address, and our lab tech will send you guys out those lab kits. I just heard from one of you, which is fine. If the rest of you can pick up your kit, that's great. But just take a look at a more previous announcement to make sure you can get to campus at this time. All right, so here we go. We're kind of in our next unit. So this chapter, and I think the next 2 will be on your next lecture exam. So we're going to start to talk about body systems, and we'll start with the muscle system. And we have all week to get through, or today and Wednesday in lecture, to get through this chapter. So we'll try to kind of break it up just right in the middle so that you kind of have shorter days in lecture. I always tell you you'll have shorter days and then you're here the whole time. But so here we have the muscle system. So we went through your skeletal system. Now we'll go through the muscle system. You guys are going over the bones still this week in lab, and the next week in lab, you'll start to learn the muscles. And we have different types of muscle tissue in the body. Skeletal muscle tissue is what's attached to your bones to help you move. Skeletal muscle tissue is striated, and that means it looks like it has zebra stripes on it, so just like stripes. And skeletal muscle is voluntarily controlled, meaning you guys control when you move your arms, when you move your biceps, your quadricep muscle, your calf muscle. So voluntarily controlled means you control it. Cardiac and smooth muscle are involuntarily controlled, meaning these types of muscles in your body will contract and move without you telling them to. So your cardiac muscle tissue is located in your heart. It's good that that is involuntarily controlled because your heart beats even when you're sleeping. So your heart muscles will contract to pump blood. Cardiac muscle is also striated, which just means it has those stripes to it. And then smooth muscle is located in your blood vessels. So it helps to constrict blood vessels when you're hot, dilates them, constricts when you're cold, dilates them when you're hot. And smooth muscle is also located in a lot of your hollow organs. So you'll find smooth muscle in your digestive tract, your stomach, in the uterus. So I think I've mentioned this before when we went over tissues, but this is why women who are in comas can still give birth, because their uterus is made up of smooth muscle, and that's involuntarily controlled. So the contractions that happen will happen without you telling them to. So here's the muscle system. Functions of the muscle system helps in movement. So think about the smooth muscle in your digestive tract. It helps to move the food and waste products through your body. Your blood vessels, the smooth muscles surrounding your blood vessels help to move blood. Your muscular system helps to maintain posture. So if you have nice kind of parallel muscles lining your spine to help you sit up straight. All of us after COVID, I think, will have this kind of Notre Dame hunchback, I think, appearance in our backs because we're all spending so much time on our computers. Your muscles help with respiration. So the most important muscle associated with respiration is your diaphragm, and it sits right under your lungs. Your muscles help to produce heat when they contract to make you shiver. Your muscles communicate with each other. They help with your heartbeat, they pump blood, and they also help to contract organs and other vessels. So that's the muscle system. Here are the properties of muscles. They're contractible, meaning they have the ability to shorten. So when we talk about muscles contracting, that means they just shorten. And when your muscles shorten or forcefully contract, they pull on the body part that they're attached to. So if you just think of your bicep, for example, your bicep is in the upper arm on the anterior side. When your bicep shortens, it's attached to your radius, so your arm will pull itself up. Excitability, meaning your muscles can respond to stimulus or stimuli. Extensibility, they can be stretched beyond normal resting length and still be able to contract. And elasticity means they're able to recoil back to their original resting length after they have been stretched. So we'll go through skeletal muscles first, also known as striated muscle because it has the stripes. Your skeletal muscle constitutes about 40% of your body weight. And skeletal muscle is named because many of the muscles are attached to your bones in the skeletal system. Some of your skeletal muscles can be attached to your skin or connective tissue sheaths, especially some of your skeletal muscles in your face that help with facial expression will be attached right to the surface of your skin to help with facial expression. Skeletal muscle is striated because it has transverse bands or striations that can be seen in the microscope. And individual skeletal muscles, such as your biceps brachii, are complete organs. So this is important to just remember that individual muscles are known as their kind of complete organ as a result of being comprised of several different types of tissue, muscle, nerve, and connective tissue. So remember, in the hierarchy scheme of organization in the body, cells made up tissues, tissues made up organs. So for something to be an organ in your body, it just has to be made up of different types of tissues. And your biceps muscle, for example, is because it has muscle tissue, nervous tissue, there are nerves running through it, as well as connective tissue. Muscles are all kind of covered with a connective tissue. And the way we kind of group muscles and break them apart, there's different types of connective tissue that will surround each kind of part within the muscle. So the first kind of connective tissue covering is surrounding the entire muscle. And this is what we call the epimysium. It surrounds the entire muscle. And then we divide your skeletal muscle into groups of muscle cells. And I'll show you a picture of this. Those are called fascicles. And each fascicle is surrounded by another connective tissue covering, and we call that the perimysium. You'll start to notice here that the suffix that has to do with muscles is my or mys or myo. You'll see that myo prefix many times throughout the body, and it usually has to do with muscle. And then each skeletal muscle fiber will be surrounded by another connective tissue covering called the endomysium. And let's see if we have a picture here eventually to show you kind of that grouping of how we break down muscles. The muscle fiber itself is a cylindrical cell. So when we talk about muscle cells, they're also referred to as muscle fibers. They have several nuclei located around their periphery on the edges. This gives you the length of them and the diameter, which you don't need to know those numbers. Skeletal muscle fibers contain several nuclei that are located at the periphery of the fiber. And then skeletal muscle cells have specific names to describe common names in other cells, for example. So the cell membrane is what we would call kind of the border of the exterior of the cell in any other type of cell in the body. But in the muscle cell, we call it the sarcolemma. And the sarcolemma has many tube-like inward folds that we call transverse tubules or T-tubules. And those are very important because when skeletal muscles contract, these transverse tubules, the T-tubules, will carry the electrical signal within that muscle to help them contract. T-tubules occur at regular intervals along that muscle fiber cell. The T-tubules are associated with enlarged portions surrounding them. They are the smooth endoplasmic reticulum, but in skeletal muscle cells, we call this the sarcoplasmic reticulum. And the sarcoplasmic reticulum is what calcium will be in. And calcium, the atomic symbol is Ca, and I put 2 pluses by it because it has a +2 charge. The enlarged portions are called terminal cisternae, and when we connect the T-tubules to the sarcolemma, to the terminal cisternae, we form this muscle triad, these three pieces. And we'll finally get to a picture soon. So here's where we talk about sarcoplasmic reticula. The big thing I want you to know about that is that it just has lots of calcium in it, and you need calcium to contract your muscles. So calcium is stored in the sarcoplasmic reticulum. You should remember that. And again, this will play a really important role in muscle contraction that we'll talk about. The cytoplasm of a muscle fiber is called the sarcoplasm, which contains lots of protein filaments. And these protein filaments are bundled up into pieces called myofibrils. We see this suffix that has to do with muscle again, myo. And then myofibrils consist of teeny tiny filaments called myofilaments. And again, we see this suffix myo in there. And the two myofilaments that make up muscle tissue are actin and myosin, and you should be familiar with those names. So let's take a look finally at a picture. So here we have, you remember, this is the structure of a skeletal muscle, and remember, complete skeletal muscles are organs. And if we look at, for example, this, whenever a muscle attaches to a bone, it does so via a tendon, which is just special connective tissue. So this is an entire whole muscle attaching to a bone, and it's covered by this epimysium. Epi, that prefix means like on top of. So epi, this is the epimysium surrounding the whole muscle. And then what we've pulled out, you can see these bundles. These are called muscle fascicles. So muscle fascicles are groups of muscle fibers, and surrounding the muscle fascicles was the perimysium, which is not labeled here, I believe. Within these muscle fascicles, they are made up of muscle fibers. And remember, when we talk about a muscle fiber is one complete muscle cell. And the muscle fibers are surrounded by this endomysium. And the prefix endo means kind of inside of. So if you get confused, epimysium is, on top of the whole muscle. The perimysium surrounds the muscle fascicles, and then the endomysium surrounds the muscle fibers as we work our way inward. The muscle fibers then, and this is a complete look at a muscle fiber. If we kind of take out what makes up a muscle fiber, you can see here that this kind of circle, all of these circles that make up a muscle fiber, these are called myofibrils. And remember, within a myofibril, we have these teeny tiny myofilaments called actin and myosin. So you can see kind of how we have the structure of skeletal muscle. You should be familiar with this, more or less what makes up this structure and how we can kind of break it down. A couple other things we'll talk about. Remember, the sarcolemma was kind of like the plasma membrane of the muscle cell. The sarcoplasm was equivalent to the cytoplasm. And then we have these specialized structures called the Sarcoplasmic Reticulum, which are all the structures that you see in yellow here, kind of. They will end in terminal cisternae. But remember, if you take anything away from today, just remember that the Sarcoplasmic Reticulum is where calcium is stored. So I'm going to just write that next to that. And we talked a little bit about the triad, which is made up of the terminal cisternae, kind of two sides of the sarcoplasmic reticulum, what they end in, and then the transverse T-tubule, which kind of are these folds that go within the skeletal muscle fiber to take that calcium or the nervous impulse down into the muscle. So now we'll talk about a sarcomere. The sarcomere in muscle tissue is the basic structural and functional unit because it's the smallest portion of a skeletal muscle that is capable of contracting or shortening. We kind of border the sarcomeres on one end to the other end by Z discs. So the sarcomere always goes from one Z disc to the next Z disc, and I'll show you a picture of this too. The Z discs will form a network of protein fibers that serve as an anchor for the actin myofilaments and separate one sarcomere from the next. And a sarcomere extends from one Z disc to the next Z disc. Then within the sarcomere, we have a very kind of unique organization of these actin and myosin myofilaments that will give your skeletal muscle tissue its striated or zebra stripe appearance. and the ability to contract. And how your skeletal muscles will contract is that these myofilaments will slide past each other. So the actin and myosin are kind of like on top of each other, like one after the other. And what they'll do, so if these are actin and myosin, they'll kind of slide past each other, and that causes the sarcomere to shorten, and that causes your muscle to contract. And each sarcomere consists of 2 light staining bands separated by a dark staining band to give it kind of a different colored appearance. We have other kind of bands, zones that are referred to with many different letters within the sarcomere. The area in the sarcomere that consists only of actin is called a lighter band because actin is thinner, so it doesn't look darker underneath the microscope. So these are called I bands. Dark staining bands are called A bands that extend the length of where your myosin filaments are. And then actin and myosin myofilaments will overlap for some distance on both ends of the A band, and this overlap causes the contraction. So we're going to talk about how this sarcomere will contract and shorten for your actin and myosin to slide past each other. And to do that, we need to talk about what actin myofilaments are made of. And there's 3 kind of important proteins that make up actin that are important. We have the actin itself, and then we have 2 proteins. One is called troponin, and one is called tropomyosin. Troponin molecules are important because they will have sites which will bind for calcium. So the calcium will come in and want to bind with troponin. So calcium comes from the Sarcoplasmic Reticulum and it will bind to troponin. Tropomyosin, these filaments will block the myosin myofilament binding sites on the actin. So eventually, for a muscle to contract, we need your actin and myosin to bind together and then kind of slide past each other. Normally, the tropomyosin is blocking those binding sites from the myosin when there isn't calcium present. So when your muscles are just relaxed, this tropomyosin blocks the myosin binding sites so that the muscle can't contract. Myosin myofilaments or thick myofilaments, so myosin is also known as the thick myofilament, they look like tiny golf clubs kind of bound together. Myosin heads, like the head of the golf club, has a place for ATP to bind. ATP is the energy that your body uses. And a really important place in your body that uses ATP is your muscles that needs energy to contract. It also includes the ATPase, which is the enzyme that breaks apart ATP, and also attachment sites for actin. So let's finally take a look at a picture. So this takes us through kind of the structural organization going from a whole muscle down to your myofilaments, actin and myosin. And then this is what a sarcomere looks like underneath a microscope. And the sarcomere, kind of the darker and lighter colors, the lighter colors that you see, that's where the actin is. And the darker colors is where the myosin is. And you can see here the Z disc is this kind of zigzag blue shaped line, and a sarcomere extends from one Z disc to the next Z disc. So this is one sarcomere, and you can see one sarcomere also kind of right here in those brackets. So within the sarcomere, we have the myosin myofilaments. They're in green, they're very thick, and then the actin myosin filaments are much thinner in purple. The I band is the area on the muscle that contains only actin. The M line goes down the middle of the myosin, and then the H zone is the area that will kind of shorten or get smaller as actin and myosin slide past each other. So you can see here kind of where my laser is, you can see, first of all, what the actin and myosin look like. The actin or the myosin has these heads and the rods, and they're bound together. And all of these heads on the myosin will reach up and connect to a binding site on the actin. And you can kind of see that up there really lightly. You can see the myosin heads kind of reaching up and attaching themselves to the actin. What these myosin myofilament heads will do is when they are attached to actin, they'll kind of like pull the actin by so that the actin will kind of slide past the myosin, and we have this shortening then of the sarcomere. The actin has kind of these reddish type proteins on it. So actin looks like a string of beads, and the troponin is this red protein, and the troponin is what will bind to calcium. So calcium, when your muscles need to contract, calcium will bind to troponin. And when calcium binds to troponin, it changes its shape and causes the tropomyosin. And the tropomyosin looks like these blue spaghetti-like strands. The tropomyosin, in the presence of calcium, will move off of the attachment sites. And the attachment sites are seen really faintly. They're these little yellow dots on the actin kind of pearls. And these attachment sites, when tropomyosin kind of slides off of them or reveals them, then the myosin heads can attach to those attachment sites and pull the actin past them. So that is really simply what happens in contraction. And we'll kind of go through those steps in a little more detail. Before we talk about that contraction, we need to talk about how your muscle fibers respond to stimuli. So one of the things that muscle fibers can do is the ability to be excitable or respond to a nervous stimulus. And in order to respond to a nerve or a nervous stimulus, they need to have an electrical charge across their membrane. We talked a little bit about resting membrane potential, and now we'll talk about that a little bit more with your muscle. So the electrical charge difference across the cell membrane of an unstimulated cell, so a cell at resting, is called a resting membrane potential. And your muscle cell fibers have a resting membrane potential, but can also perform an action potential. When we talk about an action potential, this means that that nerve or that muscle is being stimulated and it's being fired, you could say. So it means that a nervous stimulus is passing through it, and that's what an action potential is. The resting membrane potential is due always to the inside of the membrane, and it's always negatively charged in comparison to the outside of the membrane. And an action potential are due to the membrane having gated channels. When different ions pass through those channels, they will change this membrane potential so that the muscle fiber can become excitable. That's a good question, George. So an MD ordering a troponin lab test on cardiac-related issues for patients. I don't know, because we're not going to go too much into how cardiac muscle contracts, but cardiac muscle does contract similarly to skeletal muscle with the troponin. So they might, I have no idea, George, that's a great question, but what I would assume is they're checking to see if the troponin are moving out of the way so that the myosin can attach. I'm not sure how they would do that. Yeah, you should definitely ask. That's a good question. So the resting membrane potential exists because we have potassium ions are always higher on the inside of the cell, and the concentration of sodium ions are higher on the outside of the cell. They're both positively charged ions, but the inside of the cell is always more negative because it has many large negatively charged molecules like proteins inside of the cell. They're always too large to exit the cell, so it kind of keeps the inside of the cell always more negative. The presence of what we call leak protein channels in the membrane that are more permeable to potassium than to sodium allow potassium to leak out. Sodium tends to diffuse into the cell and potassium tends to diffuse out of the cell. And that's just because within your cell, there's more potassium on the inside. So potassium will naturally want to leave down its concentration gradient, and sodium, there's more sodium outside the cell. So when those channels open, sodium will naturally come into the cell. In order to maintain the resting membrane potential so that at resting it's more negative, we have what we call the sodium-potassium pump, which is one of those active transport mechanisms. And the sodium-potassium pump just recreates the sodium-potassium gradient, or the correct concentrations of sodium and potassium outside and inside of the cell, just so that the cell is always at a perfect kind of resting membrane potential so that it's easily able to respond to a stimuli when your muscle needs to contract. Troponin in your heart is a stress marker with your heart. Good. You guys are answering each other's questions in the chat. Thanks. We must have some EMTs or people who are studying that here too, or just who know this. So here's resting membrane potential and the different kind of channels that can allow the ions to pass through. So this is a muscle at rest. It's important to know the muscle at rest, the charge difference will always be more negative on the inside of the cell than on the outside of the cell. Different channels. In purple, we have a leakage channel that's constantly open for potassium to leak out. And then the other channels in pink, we have it's called a voltage gated channel. So it's closed and will only open when there's a charge or a stimulus applied to that muscle. So this is kind of what the membrane looks like at resting. So what happens? A phlebotomist, great, Cameron. So you'll know all about blood when we get there. Do you work in like a clinic or a hospital? Good for you. You probably could do my daughter's IV a lot better than they did in an ER. Very interesting. A couple of you guys are asking, so my daughter had a surgery last Tuesday to take out a pin. I should show you guys, it was about this long, very skinny. She was able to take it home. So she has this souvenir pin that was in her arm, kind of right here, and everything went fine. I think a couple of you were asking about that, but she had to go under anesthesia, which was the, probably the worst part, but she did great. And now she has a really cool pin. She took it to school and showed her teacher. So let's talk about an action potential. And this is what will initiate the muscle contraction. And what we will do is we're going to change kind of the electrical charge difference across the membrane in order to fire that cell. And when I'm talking about fire, like firing them, like starting or sparking a match. Charges in the resting membrane potential occur when gated cell membrane channels open. And when these gated cell membrane channels open, so for example, in a skeletal muscle fiber, a nerve impulse will trigger sodium channels to open, and then sodium will naturally travel down what we call its electrochemical gradient. It will naturally diffuse into the cell, down its concentration gradient toward the negative side inside the cell. Kind of like a synapse. A synapse is more referring to kind of the area when two nerves kind of come together. That's a synapse, when one nerve touches another nerve. So that starts the action potential when sodium ions rush into the cell. When that happens, remember the inside of the cell is more negative, and when sodium rushes into the cell membrane will become more positive than when the cell was at resting. And this increase in positive charge inside the cell is called depolarization. So this is kind of the first step in an action potential. We're going through this in muscle cells. When we get to your nervous system, we'll go through this in nerve cells, and the same kind of idea and steps will apply. If the depolarization changes the membrane potential to a value called threshold, an action potential is triggered. What that means is if enough sodium enters the cell so that the cell becomes positive enough, we will trigger an action potential, which is a rapid change in charge across the cell membrane so that it will go on and fire or send its signal down the entire cell. So depolarization is the first step. So depolarization during the action potential is when the inside of the cell membrane becomes more positively charged due to sodium rushing in. At the end of depolarization, all of the positive charge causes the sodium channels to close because we don't need any more sodium to rush in, and the potassium channels start to open. And when potassium channels start to open, potassium will leave the cell, and this starts step 2 of the action potential called repolarization. Repolarization means when the charge on the membrane will go back to normal, and this will occur when potassium leaves the cell. So repolarization will be due to those potassium channels opening. Potassium will leave the cell, and the outward diffusion of potassium will return the cell to its resting membrane conditions, and the action potential will end. So that's why we call it repolarizing, because we're going back to normal. In a muscle fiber, an action potential will always result in a muscle contraction. So these are kind of the steps. So the first step we saw, number one, was just resting membrane potential. If you remember the picture that looked like this. And then number two, this is kind of the first step in an action potential, is called depolarization. So what we have here, sodium channels open and sodium rushes into the cell because sodium will just be traveling down its concentration gradient. When that happens, the inside of the cell will become more positive. And that's why we call it depolarization, because we're changing the charge on the inside of the cell membrane. ECG for cardiac contraction? Yes, kind of. You guys have some great questions. Repolarization, then your sodium channels will close. So the inside of the cell has become positive. We fired an action potential, and then the sodium channels will close because we don't need any more positive charges inside of the cell. Repolarization occurs when potassium channels open. So we see a potassium channel open here, and then potassium will rush out. If you think about it, I'm just going to go back to this picture for a second. The inside of the cell at resting was really negative. And in order to become more positive, we just gained all of these positively charged sodium ions. So that's why the inside of the cell becomes positive, because sodium carries a positive charge. The reason why it goes back to being negative now is because potassium also is a positively charged ion. So if the inside of the cell is going to lose all of these positively charged potassium ions, the inside of the cell becomes more negative again. And that's how we get back to the resting membrane potential where it's negative. And we want that to be negative because when the resting membrane potential is more negative on the inside, an action potential can more readily occur again. So this takes you through just the three steps again, which ion channels open in the action potential and what three occur. So if you're still a little confused, maybe I would just encourage you to screenshot this slide and study this slide just to go through the three steps of an action potential. Sodium rushes in during depolarization, causing the inside of the cell to become positive. Then potassium rushes out during repolarization to bring the inside of the cell back to negative or resting. Okay, so a nerve supply. So muscles are supplied with nerves because they respond to stimuli. They're excitable. And a motor neuron is a nerve cell that stimulates muscle cells. We have different types of nerve cells in the nervous system, but the ones that will stimulate your muscle cells are called motor neurons. And the neuromuscular junction is the synapse. And whenever you see the word synapse, think of that as just a junction between two pieces. The neuromuscular junction is a synapse where the fiber of a nerve connects with a muscle fiber. So neuromuscular, we have a nerve connecting and a muscle connecting. The synapse refers to the cell-to-cell junction between the nerve cell, and it can either be another nerve cell or an effector cell. And in your body, nerves can connect to effector cells, and those effector cells can be a muscle or a gland. So nerves can actually stimulate glands. They can be directly connected to a gland. A motor unit is a group of muscle fibers that a motor neuron stimulates. So what that means is one motor neuron might have branches that stimulate a group of muscle fibers. And that group of muscle fibers that one motor neuron will stimulate is called a motor unit. In this kind of neuromuscular junction, we have different vocabulary to learn. The presynaptic terminal, so pre-synapse, so before the junction, is just the end of a neuron cell axon fiber, which is a part of the neuron. The synaptic cleft is the space between the presynaptic terminal, which is usually the nerve cell, and the postsynaptic membrane, which can be another neuron or it can be a muscle fiber. The postsynaptic neurons, so post-synapse, after the synapse, will be the muscle fiber membrane. And remember, in muscle fibers, we call the plasma membrane the sarcolemma. And synaptic vesicles are little tiny carrier structures that will be released from a presynaptic terminal that will release neurotransmitter chemicals. And we always need a neurotransmitter to kind of travel across the synapse to kind of continue on this nervous kind of firing from your nervous system. Neurotransmitters are just chemicals that stimulate or they could inhibit a postsynaptic cell. And acetylcholine is the big neurotransmitter that you should know. Acetylcholine is always the neurotransmitter that is associated with skeletal muscles, and it's often abbreviated ACH. So you might see it like that. That's acetylcholine. It's this neurotransmitter or chemical that stimulates skeletal muscles. So here we have the axon branch of a motor neuron. Here's the neuromuscular junction, and the axon branch could kind of branch off into different what we call terminal boutons, and each kind of terminal is called the presynaptic terminal. So what you're looking at here is the presynaptic terminal, presynapse of that axon. Within the presynaptic terminal, you'll see all of these purple little synaptic vesicles. And those synaptic vesicles are what will release the neurotransmitter acetylcholine. Acetylcholine will travel across the synaptic cleft and bind to different receptors on the postsynaptic membrane of the muscle cell. And here's a light microscopic view of a neuromuscular junction. You can see the axon branch coming down. It's kind of branching and then touching or coming very close to the skeletal muscle fiber. So that's always kind of cool, I think, to see these pictures in real life and not just what some artist drew. So you know it's actually true. Here's the function of the neuromuscular junction. It takes you through the different steps that occur. And again, just taking a step back, like what are we even talking about right now? We're trying to electrically stimulate a muscle fiber so it can contract. And in order to electrically stimulate a muscle, we need some sort of nervous stimuli. So that's why we're bringing in the nervous system. We're bringing in a nerve cell, which is showing an action potential or a signal traveling through the nerve cell. This action potential travels down through the presynaptic terminal. That action potential will open up calcium channels. And what calcium does, the opening of calcium will initiate the release of the neurotransmitter from these vesicles. So calcium is important here in the presynaptic terminal, as well as later on when we're talking about troponin. So anyway, so we have the neurotransmitter acetylcholine being released. You can see acetylcholine abbreviated there. The acetylcholine will bind to receptors on receptor molecules on the postsynaptic membrane, which is the sarcolemma of the muscle cell. When acetylcholine binds, that starts the depolarization step. So what do we remember about depolarization? The depolarization is when sodium channels open and sodium rushes into the cell. So here we have the first step in our action potential traveling along our muscle fiber. We have sodium channels opening and sodium rushing in. What this does is we have an action potential starting in one part of a muscle fiber, and then it will go in both directions to travel down the entire muscle cell. So as the action potential travels down the motor neuron to the presynaptic terminal, I'm just taking you through the steps that the picture just showed. It causes calcium channels to open. Calcium causes these synaptic vesicles to release acetylcholine. Acetylcholine travels across the synaptic cleft, which is the space between the two. It binds to receptor sites on sodium channels. Sodium channels open, sodium rushes into the postsynaptic terminal, and we get depolarization, the start of the action potential. So we're starting to excite or electrically stimulate now our muscle cell. Now we get to the fun part. So sodium causes sarcolemma and T-tubules to increase the permeability of sarcoplasmic reticulum, which will release stored calcium. So in that location where depolarization occurs, that will cause the sarcoplasmic reticulum, which again, hopefully you remembered, that's where calcium is stored. So the depolarization causes calcium to be released. And this is when we get into the steps that we talked to a little bit before. Calcium then binds to troponin, which is attached to actin. Calcium binding to the troponin causes those spaghetti-like strands called tropomyosin to move or slide off of the actin, the myosin binding site. So they will expose the attachment sites for myosin. This will cause your myosin heads, they look like a head of a golf club, to bind to the actin. ATP is released from the myosin heads, and the heads will kind of bend towards the center of the sarcomere. And this bending, if the heads are bending, but they're attached to actin, it will just slide the actin past. Acetylcholine esterase, if anything ends in ACE, that's an enzyme. This breaks down excess acetylcholine in order so that your muscles aren't being constantly stimulated. Remember, we need acetylcholine to start depolarization. Acetylcholine esterase kind of controls the amount of acetylcholine just so that we're not constantly contracting that muscle. Sodium channels will then close and the muscle contraction will stop. So here we have kind of the steps in order that I'll let you guys read through. Action potential traveling along, calcium channels opening, causing the release of acetylcholine, which attaches to your sodium channels. Sodium then enters the cell, causing depolarization. And when the action potential kind of jumps down, and that's what this dotted line shows, this dotted line is showing kind of the generation of the action potential down the length of a muscle fiber cell. It will travel down into the T-tubule, and it'll cause the Sarcoplasmic Reticulum, which is in yellow here, to release calcium. And calcium is very important. When calcium is released, it can bind to the troponin, which causes the tropomyosin to expose the myosin binding sites. Myosin can then bind to actin and cause it to slide past each other. And that's what this picture shows, the calcium binding to troponin in red. When that happens, the tropomyosin, which are these blue spaghetti-like strands, they've moved out of the way, and you can see here in yellow each of the myosin binding sites. So these yellow kind of places will be where the heads of those myosin filaments will bind. ATP kind of has to be broken apart for the myosin head to bend, and that bending causes the actin to slide or move. So here again, these past 5 slides are actually great because they show pictures and take you through step by step. So when you're studying, I'd encourage you maybe to look at these slides individually because I think they're really helpful with how they go through it. So here we have the heads of the myosin myofilaments bend, causing the actin to slide past the myosin. And as long as we have sufficient calcium present, the cycle will repeat itself. So where do we need energy? Where do we need ATP? And why is that important for muscle contraction? And we'll probably end in a couple of slides here. We're almost halfway through this chapter, and we'll leave some fun for Wednesday in lecture. So we need energy, and energy for muscle contraction is supplied by adenosine triphosphate. ATP, again, is your body's energy currency. It's like the money that your body needs to do work. So this is ATP. Energy is released as ATP because when we break ATP apart, when we break the bonds and we break it down to adenosine diphosphate and a phosphate group, when we break a bond, we release energy. ATP is stored in those myosin heads, and ATP helps to form the cross bridge formation between the myosin and the actin. This is what we're getting at here. New ATP always is required. It must bind to the myosin before the cross bridge is released. So what this means is that your body or your muscles will maintain a constant contracted state unless new ATP is present, because new ATP, if this is the myosin head and this is your actin, it will maintain a connection, a contracted state, until more ATP comes to allow the release. So this is where rigor mortis comes in. Rigor mortis means stiffening of death. So rigor mortis will occur when a person dies and no more ATP is available because the body cells are not creating ATP to release the cross bridge. So in death, you can kind of count or calculate how long a person has been dead by looking at how rigor mortis has set into their muscles or how stiff their muscles are, because the muscles need ATP to kind of detach these cross bridges. Right after death, the muscles will maintain in this contracted stiffening state. Eventually, and I don't remember the specific number of hours, I think for sure 12 hours after death, eventually the muscles will just start to degenerate. So eventually the muscles will relax, but rigor mortis is the stiffening of the muscles right after a person dies. So here's the ATP breakdown and the cross bridge movement, showing again how calcium comes in, exposes the sites so that the head of the myosin can bind. The myosin heads bind to the sites to form what we call a cross bridge, which is just a bridge between the actin and the myosin, shown here. Energy stored in the myosin head is used to move the myosin heads, and that's what we call kind of the power stroke or the bending. And then extra ATP is required down in this step to remove the head from the actin. And right here, this is the step your muscles kind of get stopped in when a person dies. The myosin head is just bound to the actin, and not everywhere in the body, but I mean, your body's muscles are constantly contracting. So some muscles will be stuck in this state at death because energy is needed to detach this cross bridge, and the cycle will just continue. So I think let's get through twitching and then we'll take a stop there. So a muscle twitch is just a single contraction of a muscle fiber in response to a stimulus. And a muscle twitch has three phases: the latent phase, the contraction phase, and the relaxation phase. The latent phase is the time between the application of the stimulus and the beginning of the contraction. So right when you stimulate a muscle and right before it contracts. So that is a very short stage. The contraction phase is the time during which the muscle is contracting or shortening. And then we have a relaxation phase. So here we have a stimulus being applied. The lag this is the latent phase, and then the contractant phase and the relaxation phase. So this is one single muscle twitch. The y-axis shows the tension of those muscle fibers. The x-axis just shows over time. Summation and recruitment have to do with how muscles can kind of contract more forcefully. So in summation, individual muscles can contract more forcefully. Tetanus is when we call a sustained muscle contraction, that it occurs when the frequency of stimulation is so rapid that your muscles never relax. And it's never good for your muscles to be in tetany because they're constantly contracting and never relaxing. Some drugs, some toxins can cause tetany of your muscles. Recruitment is the vocab word we use to describe stimulating several motor units. And this can occur if you guys are running a race, if your legs are doing hard lifting work, your body will recruit several motor units to go to your quadricep muscle to try to increase contraction to do that work. So here we have in a graph format what these different kind of twitches look like. So here is kind of a twitch with a frequency stimulating that muscle. Here's incomplete tetanus, kind of shown up here, where you'll have kind of a contraction phase. And remember, the relaxation phase is when it goes down. So we can have incomplete tetanus where the muscle is still being contracted, but it's given a little bit of a relaxation phase. And then complete tetanus just means we are in a constant state of contracting. Tourette's, a muscle twitch. Yes, I would have to look more into that kind of a constant twitching. Yes, but not necessarily the muscle twitching is causing the Tourette's, but that it's from kind of another imbalance in the body. Good question, Kara. I'll take a look at that when we're done. Okay, so we will stop there and we'll kind of finish up on Wednesday, starting with skeletal muscle fiber types. And we made it a little over halfway through. So I'll go ahead and stop the